Assalamu alaikum students. I hope all of you are in good health. Welcome to my course Introduction to Biotechnology and Genetic Engineering. This is our second class. And from previous lecture, we get to know the basic terminologies related with biotechnology. And of course, we have seen the significant discoveries of some renowned scientists. Among them, we see what Louis Pasteur did. He invented pasteurization and also he first commented on that microorganisms are responsible for fermentation. Later on, we saw that Pasteur also discovered rabies vaccine. And then comes Charles Darwin. Charles Darwin have focused on natural selection procedures to give us a theory of evolution. Later on, we move towards the father or the founder of genetics, Gregor Mendel, whose laws are most important statement in the era of modern biotechnology or modern genetic engineering. The two laws of segregation and independent assortments, were the root for knowing the pattern of heredity, for knowing that why an offspring or a child should have certain features coming from its parents and other relatives. After that, we know about Alexander Fleming, who first discovered penicillin, and Robert Koch. The invention of Robert Koch was the initial stroke in modern microbiology. But as our course is related with genetic engineering, we need to have some basic idea about genes and other basic molecules. Here in this lecture, we'll know more about some other definitions, and this lecture is very important or a baseline for learning biotechnology and genetic engineering, and of course for all other sectors of biological science. That means here you will learn about some terms or terminologies or some mechanisms, some sort of pattern that will help you to have a brief idea about every sector in molecular biology or basic biochemistry. So let's start. Gene. As we are trying to understand genetic engineering procedures, we first know what is a gene or what is the genetic material. By this time, it is clear that gene is our genetic material that help us to carry the function or to carry the information of our parents. It is the gene that will determine your behavior, determine your structure, your anatomy, your everything. Your mood swings, from your mood swings to other patterns, your speaking style, your writing skills, and everything will be determined by the function of this gene. So what is this gene? This gene is basically the segment of a DNA. That means we found DNA within chromosome and we found gene within DNA. Please try to correlate among them. Gene is integrated, or gene is a portion of DNA, not the whole DNA is called gene. Please do not confuse with it. The whole DNA is not gene. There are thousands of genes present in a DNA. Alright, thousands of segments of DNA are called gene. So this gene has the information for protein synthesis or RNA synthesis. We will learn both about protein synthesis and RNA synthesis in our upcoming lectures. This single molecule of DNA has thousands of genes, as I mentioned just now. Now there is a mantra or there is a dogma that is called central dogma. And the central dogma said from DNA we can have RNA. That means DNA is converted to RNA and further RNA is converted to protein. And this is the basis of all types of molecular biology. In every aspect, we need to convert a DNA into protein in order to see the ultimate outcome or the ultimate result. Suppose you are modifying a certain DNA of a certain organism. Now you need to know whether your modification has been done properly or not. How can you analyze this? Of course, you can analyze this by applying some molecular biology approach, but the ultimate result will be determined by the outcome or the phenotype of that particular organism. For example, like you have changed a certain gene of a rabbit in such way that will give the rabbit a different eye color. In normal case, the rabbit might have a blue eye, or a rabbit might have blue eyes, but you have changed its gene for eye color, its gene for that are responsible to give its specific eye color in a way that will further give it a green color eyes. Now, how can you be sure that whether your this modification or whether your this genetic engineering procedure has been done successfully or not? By seeing the ultimate phenotype. This is the protein, the ultimate phenotype. When the gene or the modified gene will express and form protein, and this protein will finally lead the rabbit to have green eyes. And this is the basic theme of running any genetic engineering or molecular biology experiments. So this is the basic concept of gene. Gene is a portion. Portion means a fragment or a segment of DNA that is information, and this information is passed towards RNA and finally in the DNA. And another thing, important point is that a single molecule of DNA has thousands of genes. Not the whole DNA is called a gene. A DNA can carry more than thousands of genes. That means, for example, human DNA has approximately 25,000 of genes. We'll know later that how these 25,000 of genes will nurture all our behavior and all our functionality. Now, this figure is the most important figure. This might look a bit clumsy, but I can assure you that this is a very easy figure. Just listen to me carefully. What is the title? The title of the figure is Chromosomes are composed of DNA and nucleoproteins. This is the basic information from this figure. That means chromosome. Chromosome, here in this figure you can see this is the whole cell. This is the nucleus, and in the nucleus there are so many chromosomes. 1, 2, 3, 4, 5, 6, 7, 8, 9. Almost 9 chromosomes. And this is the chromosome in a clear view. This is the chromosome. And if you magnify it in a larger with a magnifying glass or a microscope, then you can see within this portion, this small amount of portion of chromosome, this whole thing is compacted. Now let's see how chromosome is composed of DNA and nucleoproteins. This is the DNA. I think all of you know the structure of DNA. This is DNA having 2 helical structures and base staircases, just like a twisted ladder. Alright, and these spherical particles are proteins, or specifically histone proteins. Alright, these are proteins, and this blue colored thread is the DNA, and this is the elaborated or expanded view of DNA. So this DNA is wrapped around these histone proteins. In one sentence, the histone proteins will provide a platform or a scaffold for DNA to form a solid structure. Just imagine you are wrapping a scotch tape around a tennis ball. Most of the baby boy do this too for their playing purpose. Just like that, when you are wrapping a specific scotch tape around a tennis ball, here the tennis ball is similar to this histone proteins, and the wrapping scotch tape is just like the DNA. So the DNA is wrapping around these histone proteins, forming small beads. Like this is 1 bead, 2 bead, and this is 3, 4. There are so many of beads, and these beads are finally linked with a linker DNA. This is also a DNA. This is also a DNA. This linker DNA will connect both the beads and finally push them together to form a compact structure like this. This is coiling. This is coiling of DNA around a histone protein and finally forming a beads. And this bead structure is called a nucleosome. Alright. So what is the definition of nucleosome? The definition of nucleosome is the structure that is formed by wrapping around of DNA, by wrapping of DNA around a histone protein. So then these nucleosomes will come together or push together by these linker DNA and will form a more compact and more coiled, or this is called highly coiled structure, just like this. And finally, this coiled structure will compact more to have a supercoiling. You can compare this with a tangled telephone wire. Some telephone used to have some kind of wire in ancient period that was analog telephone. Not just like today's digital telephone. It was analog telephone where we have to use a spring type of number plate to dial, and there are this type of supercoiled wire. So what we have seen so far now, it is the DNA that is wrapped around these histone proteins forming nucleosome structures, and these nucleosome structures finally come together to have a more complex structures that again push together to form a supercoiled structure, and this supercoiled structure were compacted together to form the chromosome. Just imagine you are pressing something and forming supercoiling. That is why the diameter or the total ratio of that particulate will decrease in such way that you can incorporate it in any kind of smaller hole or smaller pocket. This is the nature of, or this is the pattern of chromosome packaging. Now let's see. This is DNA and this is nucleoprotein. As this protein only found in our nucleus, that is why these are called, or these histone proteins are called nucleoproteins. Later on in this lecture, we'll also see another type of nucleoprotein called histone protein. So these nucleoprotein and DNA together form the structure of chromosome. For better understanding, I will also record this lecture in Bangla and also provide you with supplementary videos in the website of your university so that you can have total access of those. So now that we know what is chromosome packaging, let me show you what is the basic construction. So DNA and proteins associated with the DNA, that means proteins associated with the DNA ultimately form the chromosome. Histone proteins help DNA coil up and form its shape. So histone proteins have some structural function on chromosome, and there another type of protein called non-histone protein. Please keep in mind that non-histone protein has no structural function over chromosome. It just has some regulatory functions such as turning on and turning off of a gene. Now you might have a question, what is turning on and turning off of a gene? Well, this simply means whether a gene will express or gene will convert it to protein or not. If it is enforced to convert into a protein, then we can say the turning on of gene, and if it keeps silence without forming any protein, then we can say turning off of genes, and this is regulated by non-histone proteins, and all of them were found in the nucleus.